Welcome back. This is almost towards the end of this part of the study design. It's not very big, this section, no, not in 2020. They've cut out a lot. So the study design specifies that you must know the use of phylogenetic trees to show relatedness between species. So we're just going to show you a couple of ways that you can do that. Now, these notes have differed quite a little bit from what you've got. So if you can go to about page 6 and 7, that's where we're going to be working through. But I've actually added another slide about molecular homology trees. So you might need to add some information in somewhere on one of those pages. So let's have a look. Let's get started. So what are phylogenetic trees? So this is at the top of page 6. So these are any kind of branching diagrams that can depict evolutionary relationships between organisms. They give us a sense of when things diverged and which things are more closely related to each other. So previously we built them on differences between physical characteristics, and that's what we're going to focus on first, which is in your notes. And now they use molecular homology to determine how related two species are to each other. And we're going to look at some benefits and disadvantages for those. So we're on page 7 now, but you'll notice that I don't have a little subheading called benefits. So you might have to add that in. So these are the historic trees. So you might have seen them. They look a bit like this. This is what I call a comparative morphology tree. So this is based on what they look like. Yeah, it's based on physical similarities between organisms. And we've determined that, say, the sirens, which are things like dugongs and the elephants here, have the most physical features in common. So therefore we put them very close to each other with this one little branch. So that's down the bottom down here. Whereas things like monotremes, which are your platypus, versus the primates, which are your monkeys, they diverge from each other all the way back here. Yeah. So the closer this is to the front, the closer this is to the front. I'm going to call this the front. Yeah. So the closer to the front equals a more recent common ancestor. Yeah. But what we've done is over time we've built these trees based on how different things look to each other, their physical similarities. Now, the good thing about this is it's very fast. You don't need any special equipment. However, you've got to think that over time, organisms may have lost some of their features. So think about a whale. Yes, a whale lives in the ocean and it doesn't have back legs, but it's actually very closely related to an elephant or to a hippo. Whereas you might think it's more closely related to a shark. Yeah, so analogous characteristics here make it difficult. So organisms that have gone through similar selection characteristics, there we go. Organisms that have gone through similar selection pressures and developed similar physical features can be placed on a tree much closer to each other than they should be. Yeah, because they aren't actually very similar to each other when you look at their genetic makeup. So analogous features will cause species to be thought of more closely related to each other than they actually are, as they look similar. And the example here would be something like a dolphin or a whale and a shark. Yeah, they're not very closely related, but if you just went on physical features, you would say, yeah, they do look like they could be very closely related because they have very similar body shapes and so on. All right, so what we use these days, and this isn't in your notes, so you will have to probably pause and write this in. So what we use these days is we use a molecular homology tree. So this is based on the differences between the DNA or the amino acids. So this is what we were talking about in the last video or two ago. This is molecular homology trees. So these are not affected by analogous features. Really good. They also can show similarities if you look completely different. So a whale and a bat, you'll find out they're way more closely related than a whale and a bird, or a bat and a bird. Yeah, because it's all hidden in their DNA and hidden in their protein structure. Now, there are a couple of limitations, but to be honest, compared to a comparative morphology tree, these limitations are very small. So sometimes things can mutate twice in the same spot. And what that will mean is that there's actually been twice the mutation rate, but you can't see it because it's happened twice in the same spot. Yeah, it's like writing over someone's VCR tape and then writing over it again. You wouldn't have known that there was that material there in the middle. Yeah, so I know I use VCR. It's a really bad example, but hey, I can't think of another one right now. The other thing about this is it's more expensive. obviously, and more time consuming. But we are doing this way more often than others. So what I'm going to do now is I'm going to go through a few trees, and then I'm going to go through what they're talking about on the news at the moment, which is the genomic sequencing of the COVID outbreak. So let's just have a look. We've got two different phylogenetic trees here. This first tree was done using what we call morphological data or physical similarities. Yeah, so this was the comparative morphology tree, and this second tree was the molecular similarities. Now, what you'll notice is that they look completely different. Yeah, the only thing that's similar about them is that we understand that both monotremes and marsupials are very kind of distantly related to the rest of these mammals. Okay, so all of these groups are the mammal groups, mammal families, or mammal, I think they are mammal classes. No, mammal orders. There we go. We'll get there. That thinking about a kingdom, phylum, class, phylum, class, order. Yeah, order primates. Sorry, kingdom, phylum, class mammalia. Orders. These are all the different orders. They're the one underneath the class. So here's mammals, and now we're looking at all the different orders of mammals. My God, I'll get there. All right, so what you can see is that if you look at genetic differences between them, that these primates, for example, the most closely related group to us, so the way to figure out your most closely related group, and I'm going to clear my pen markings and we'll just start again. So I'm only going to use this molecular tree, and we want to figure out what's the most closely related group to the primates. So what I do is I go back in time to where they branch off, which is here, and then I come out again, and I say, all right, the two most closely related groups to that primate are the Dermatoptera, Dermoptera, there we go, and the Scandentia. So these are the ferrets and what looks like flying foxes. Yeah, so they, out of all these classes, they're the most closely related to us outside of other apes. So if you go to other mammal groups, they're the most closely related. Let's have a look at another species. So let's say, okay, I want to figure out, is the, here we go, we'll go to the sirens again. So is the dolphin more closely related to an elephant, or is it more closely related to a primate? Okay, so what I'm going to do is I'm going to find all three. I'm going to find my primates. I'm going to find, hang on, let's just get rid of these pen markings. So here are my sirens. Are they more closely related to the elephants, or are they more closely related to the primates? Now what I need to do is I need to follow back my chains until they join at a common point. So having a look, let's do one in green. So let's do the, what's that, an elephant and the siren in green. So what I do is I just go back, and I go back and I find this point here, okay? So this green point is their most recent common ancestor, yeah? And if I want to compare the sirens, which in yellow, sorry, in orange, to the primates, I have to go all the way back, this one too, all the way back to here. They have a less recent common, and this is the language you're going to use, yeah? Less recent, more recent, less recent or more recent common ancestor. So I would say that the dugong is more closely related to the elephant than it is to a monkey or a human, because they have a more recent common ancestor, okay? So we are more closely related because they have a more recent common ancestor. So you should be able to do that on just about any tree. You would say here that the lizards are most closely related to the mammals because they have the most recent common ancestor. The lizards on this tree are the least, let's just write that here. So lizards and sharks are the least related, as they have the more, so you could say, let's just keep the language the same. You could say as they have a less recent common ancestor. So like I've told you, everything's got to do with directional words. So in this case, the directional word is how recent was that common ancestor. Here's another tree. Yeah, you can determine that the parvulus is more closely related to the pallidus than it is to the Fusca, because the Fusca's common ancestor is all the way back here. Okay, so these ones here, these two here, are more related, most related out of those three, because they have a more recent common ancestor. Here is their common ancestor. Here is the different, this is the SARS virus. And what we found is that this is the strain that infects humans. The one that is most similar to that is one that infects civets. So they're like these kind of little cat-like creatures. So what that says to us is that actually the strain of SARS that hurt a lot of humans, or that infected a lot of humans, must have stemmed from a strain that had mutated to infect these civets. So the virus that you find in the civets is more closely related, has less genetic differences to the human virus compared to the ones that infect bats. So that's pretty cool. So COVID genetic sequencing is a little bit different. It's a little bit huge, but this is real data, yeah? Real data from the beginning of last December to this April. And what it's showing you is that it started off with a strain kind of back here, this purple strain, yeah, somewhere, this would have been probably in Wuhan. And then there were 4 kind of main branches that came off from that. And then you can see that quite a few of them died out, yeah, these different strains. Now, the good thing about a virus is a virus mutates quite frequently, because it's got that high reproduction rate, and every time it reproduces, it's got a chance of mutating. So you can see that some of these dots and some of these branches actually die off, which means those strains have been eliminated, and there would be some more mutations over here. So what you would say is that these viruses here have a common ancestor back here, yeah? But if you wanted to compare them to these viruses down here, their common ancestor is all the way back in Wuhan. They must have split off there. And so what they can do from that is they can actually construct some pretty cool diagrams to show us where the virus is spreading from and to. So you can see back in January, a large purple dot showing that it originated in Wuhan. Then it moved to America and it mutated, and that's the red. Yeah, is that mutation in America. And then you can see the American dot actually going and infecting a lot more places, whereas that purple Wuhan strain actually has now disappeared almost. And that's only in March. So it's these mutations that can give us a kind of a map as to where the virus has come from and where it's spread to. So here, for example, is a very small part of that big phylogenetic tree. And what we can do is they can collect random genomic testing from people that have COVID, and they can determine which strains are likely to have branched off from others. Yeah. And so that's where articles like this one come into play. So this one was on August 17, so not that long ago. And what it showed was that 99% of the second wave in Victoria was caused by a new strain of the virus coming into Australia from returned travellers. So it wasn't going to be a strain of the virus that had just existed in our community before then. It was a breach somewhere where we got returned travellers that have managed to spread their infection in the population again. So it wasn't the same strain for this second wave compared to the initial wave, which is really, really interesting. And I'm happy to have a conversation with you about that later on too.